Hello everyone, welcome to my channel Anirvan Science Fair, where we explore, discover and learn new concepts. Here today we are going to deal with oogenesis. So let us begin our understanding with the primary oocyte. The story begins. In case of humans, including human females, primordial germ cells, that is PGCs, do not form within the developing gonads, that is the ovaries themselves. Instead, they are formed outside the embryo during early development and are first identifiable within the wall of the yolk sac near the base of the allantois around the 3rd to 4th week of gestation. From this location, the primordial germ cells migrate along and finally reach the gonadal reaches, which ultimately develop into ovaries during the 5th to 6th week of development. And once the primordial germ cells enter inside the ovary, they differentiate by mitosis. So oogenesis is the biological process of formation of female gametes within the ovaries. Unlike spermatogenesis in males, which is continuous from puberty onwards, oogenesis is a discontinuous process that begins before birth, pauses for years, and ends with menopause. It is highly regulated and complex, involving the processes of mitosis, cell growth, and 2 stages of meiosis. So now we are going to talk about the stages of oogenesis. The entire process can be conceptualized in 4 major phases aligned with human life cycle stages. Phase 1, it is a prenatal phase, that is before birth. Phase 2 is puberty, in which there is cyclic activation. And phase 3 is ovulation and fertilization. And phase 4 is completion and fertilization. So phase 1, prenatal phase, that is the phase that takes place before birth. This phase establishes the total pool of germ cells for the woman's lifetime. Migration and mitosis. The primordial germ cells, which migrate from the yolk sac to the developing gonadal ridge around the 5th week of gestation, once there, they differentiate into oogonia. And then there is proliferation of the primordial germ cells. That is, the oogonia are also diploid stem cells. They undergo extensive mitotic division to multiply rapidly. By the 5th month of gestation, there are millions of oogonia in the ovary, around 7 million. But many of these begin to undergo atresia, that is natural cell death before birth. Then starts the process of meiosis. A subset of the oogonia grows in size and begins meiosis 1, and they are now termed as primary oocytes. Now, arrest in prophase 1, that is the diplotin stage of prophase 1. Crucially, the primary oocytes do not complete meiosis 1. They stop at prophase 1, specifically the diplotin stage, and they are housed within a primordial follicle, that is a single layer of flat granulosa cells surrounding the oocytes. This is the state of all potential eggs in a girl at birth. Then phase 2 starts with puberty. So after birth of a child, till she reaches puberty, nothing happens. The primary oocytes remain in a suspended state. When puberty is reached, then Following the resting phase of childhood, puberty initiates the cyclic reactivation of a set of primary oocytes, and folliculogenesis occurs. In response to the follicle stimulating hormone, that is FSH, secreted from the anterior pituitary, a cohort of dormant primordial follicles begin to mature each month. They progress to primary, secondary, and finally a single tertiary follicle, which is also known as Graafian follicle. As the follicle develops, the primary oocyte within it also grows. And after the Graafian follicle is formed, once it is completely matured, then due to the surge in luteinizing hormone from the pituitary gland, the pre-ovulatory surge, which is called, it signals the large primary oocytes in the Graafian follicle to restart meiosis 1. Phase 3, ovulation and fertilization. Asymmetric division of meiosis 1. This is a vital difference from spermatogenesis. During the completion of meiosis 1, the primary oocyte, which is diploid, divides the cytoplasm very unevenly. One cell retains almost all the cytoplasm and nutrients, and the secondary oocyte N is formed. This cell has a 1N ploidy but 2C DNA, 2 sister chromatids per chromosome. The other cell is very small with very little cytoplasm, and this is called the first polar body, which is also N. It is essentially a genetic trash bin, ensuring the egg becomes haploid. The polar body often stays within the zona pellucida and eventually degenerates. Ovulation and arrest in metaphase 2. The secondary oocyte is expelled from the ovary, and that is called ovulation, into the fallopian tube, and it simultaneously enters meiosis 2 but is immediately arrested at metaphase 2 stage, and the ovulated egg is actually a secondary oocyte, and it remains in this state only for about 24 to 48 hours. Phase 4, completion and fertilization. The second meiotic division is conditional. It only happens if a sperm successfully penetrates the secondary oocyte. Fertilization and completion of meiosis 2. When a sperm fertilizes the secondary oocyte, the influx of calcium ions triggers the rapid completion of meiosis 2. And in this process also, a second asymmetric division takes place, and another extremely uneven cytoplasmic division happens. As a result, another polar body is formed, which is called the second polar body. The large cell that retains most of the cytoplasm is the otid, and a second body, which is smaller in size, that is expelled, is the second polar body. Sometimes the first polar body also divides into two, but usually all polar bodies degenerate. The last phase, that is phase 4, completion and fertilization. After fertilization, the nucleus of the otid fuses with the nucleus of the sperm to form a single diploid cell, that is the zygote. And if fertilization does not occur, the secondary oocyte will never finish meiosis 2 and will degenerate within the fallopian tube. It is eventually sloughed off during menstruation.
Now let us look into the hormonal regulation in females during oogenesis. The anterior pituitary secretes FSH and LH. FSH helps in follicular development and estrogen secretion. LH helps in ovulation and corpus luteum development. The corpus luteum releases two hormones, estrogen that helps in thickening of endometrium and progesterone that maintains endometrium.
Thus we can say that oogenesis is a remarkably choreographed process of discontinuous growth and division designed to maximize the developmental success of a single well-provisioned gamete. The significant contrasts with spermatogenesis highlights the divergent reproductive roles of the two sexes, where the male specializes in producing immense number of motility optimized gametes. The female focuses on producing a few exceptional nutrient rich gametes that can support the start of a new life.
Thank you for watching this lecture. Hope it was helpful. Please like, share and subscribe to my channel Anirvan Science Fair.